The future of science is very small. I am Dr. Soumendra Darbar, faculty member, Department of Life Science, Jadavpur University, India. Today, the title of the talk is A Novel Grain Synthesized Silver Nanoparticle Act as a Nanozyme and Potentially Useful Against Oxidative Stress, Challenges and Opportunity. Biomedicine, especially nanomedicine, is the medical applications of nanotechnology, the promising weapons in the medical world to treat various communicable and non-communicable diseases. This target-specific medicine is very useful for prevention and treatment of various life-threatening diseases through inhibition of ROS, that is reactive oxygen species. We know various ROS molecules like peroxides, hydroxyl ion, hydroxyl radicals and superoxide anions. When the produce of these types of ROS molecules, a huge number within the body, it produces deleterious effects towards the cell. It is observed that liver disease is the fifth most common causes of death. Every year, 400 million detected causes potentially life-threatening liver diseases. Hepatic fibrosis is a particular disease related to liver, generally developed through oxidative stress. Still now, there is no specific drugs available in the market, particularly treat for liver diseases. There is no phytometallic nanoparticles that can be used orally against chronic diseases because of their non-specific distribution to the entire body. So, there is a question. Is there any oral treatment against the chronic liver diseases? Or is there any treatment to prevent the oxidative stress? Our aim and objective of the work is testing the potential of silver nanoparticle coupled with andrography speniculator as therapeutic agent against chronic liver diseases, particularly in the liver fibrosis. So here we all know that about a synthetic drug have numerous drawbacks like reduced bioavailability, non-specific distribution of the entire body, low retention time and undesired adverse side effect, huge adverse side effect. That's why scientists all over the world nowadays engage to develop nanomaterial, which is a target specific materials and have a very potential effects towards the cell like transport across biological cell membrane, high specificity and bioavailability, high reactivity due to higher surface to volume ratio, the simultaneous diagnosis and therapy, and the optical, electromagnetic and biological property. Still now, US FDA approved 51 nanomedicines and 77 products are in clinical trial. Here, synthesis and characterizations of the silver nanoparticles coupled with andrography speniculator. This is the synthesis procedure. Andrography speniculator reacts with silver nitrate and produce nanoconjugate. This is the characterizations of silver nanoparticles. We confirm that our developed product is being nanoformed through the spectroscopic study, TAME, XRD and FTIR. This is the in vivo treatment protocol. We choose 4 groups. Group 1 served as a vehicle control, group 2 CCl4 treated, group 3 CCl4 plus our developed nanoparticles, and group 4 that is only nanoparticle treatment. We studied various types of parameters like alpha-SAB, hematoxylin, eosin, various ROS enzyme, various liver function test enzymes, etc. So here is the morphometric parameter like body weight, food consumption, water consumption, and we noticed very positive effect after applications of our developed nanoparticles. Now this is also some morphological and biochemical observation like liver weight, liver body weight ratio, body fat. All these are the morphological parameters, and burn, total bilirubin and direct bilirubin is the biochemical observations. In the group 3, we notice the deleterious effect of CCl4 upon the liver disease, and in the group 3, that is CCl4 plus nanoparticle therapy, that restored the deleterious effect which is observed after treatment of CCl4. So this slide also stated the hematological observation. This is the control group, this is CCl4 treated group, this is CCl4 and nanoconjugate treatment, and this is nanoconjugate treatment alone. We observe that the RBC levels, WBC level, platelet and hemoglobin shows the positive protective effect when we treated with developed nanoparticles. So here, very interesting, some Roche related parameters like catalase, GSH and GPX. We measured both the liver ROS and both the serum ROS, and both the study showed that the group 3 produced significantly positive effect upon the animal models. So here is the another parameters, some different parameters, MDA, ROS, total ROS contain and superoxide dismutase. We also observe the same positive effect after treatment with our developed nanoconjugate. Now here some very interesting, the immunohistochemical study. Normal liver treated with CCl4 produce liver fibrosis. When the liver fibrosis is developed and we apply developed nanoconjugate in a certain time period, this fibrosis is almost recovered and it developed the healthy liver again. So here is the immunohistochemistry of alpha SMS. This is the control group. This is the CCl4 treated group. Clearly shows the deleterious effect of CCl4. This is the CCl4 plus nanoparticles, 50 micrograms. It shows there is positive recovery, and the nanoparticles also, only nanoparticles, it shows there is no toxic effect on the nanoparticles. This is the histological scoring. The second group shows the significant deleterious effect, and this is almost 90% recovered after applications of the developed nanoconjugate. Here the special staining, Passon trichromes, we also the same positive effect after applications of the nanoconjugate, and here is the DNA fragmentation study. Same thing, the positive effect observed in the group 3 where we treated the nanoconjugate. So The summary and conclusions of the study: we developed silver nanoparticles coupled with andrographis paniculata. The process is eco-friendly, cost-effective, and industry-friendly. So after applications of silver nano conjugate, we observed a promising antioxidant activity which can be very useful in development of the new drug for biomedical applications. This biological chemistry approaches towards the synthesized silver nanoparticles through green route is highly essential effort being addressed in nanomedicine because of its varied advantage as a future medicine. So here are the challenges of the biomedical research, like large animal experiment, stability study, details clinical study, bioequivalence study, some sponsor pilot scale study, some regulatory licensing, safety and efficacy study, marketing, and also the advertisement. The opportunities of the biomedical research in the newer after post-COVID era: it is safe and symptomatic medicine, very low cost, target specific, non-toxic, easy to produce, high efficacy, and if we market the nanomedicines, it produces royalty. So biomedicines, the nanomedicine is the future medicine. So we thankful to our teacher, Professor Somir Kumar Pal, Dr. Atish Kumar Chattopadhyay, Dr. Ranjan Das, Dr. Siddhartha Bhattacharya, Dr. Devashish Pal, my senior colleague Nabarun, all the funding agency, DST and DBT, all the lab technicians, and especially the faculty and staff members of SN Bose National Center for Basic Sciences for giving the financial support as well as the instrumental and lab facility. Thank you all.